Spatial transcriptomics is becoming an essential data type to include in any study related to the cellular and molecular function of tissues. However, the analysis of spatial transcriptomics is complex, often requiring the help of a bioinformatician who may not know the biology of the system. The SpatialGE-Web app has been designed to help tissue biologists with data analysis of spatial transcriptomics. SpatialGE does not require experience with coding or programming and allows users to do exploratory and advanced analysis that can lead to hypotheses to be tested in the laboratory. SpatialGE can be accessed from its website, spatialge.moffitt.org. Once there, users can create an account or log into an existing one. Step-by-step documentation is available for each module. Example datasets for demonstrating SpatialGE's capabilities are also available. Metadata, such as clinical outcomes or properties of each tissue sample, can be incorporated into the pipeline. Users can create filters for spots, cells, or genes by adjusting options in the interface. To help assess quality control and results, plots are generated at each step. Data and associated results are securely stored within the user's account. Options for data normalization are available, including SC transform. SpatialGE-Web app can be closed and processes will not be stopped. Users can choose to receive an email notification when the analysis is completed. Modules on the left sidebar are enabled as the user completes pre-requirement tasks, guiding the user through the pipeline. Principal component analysis can be performed to assess batch effects or biological patterns. Users can also check the number of total counts or genes per spot or cell. The visualization module generates high-quality plots of the expression at each spot or cell. The layout facilitates comparison across samples and genes. Spatial interpolation or kriging is available to create gene expression surface plots. Comparison with tissue images, if available, is also facilitated. Plots can be downloaded as PNGs or as PDFs and SVGs, editable with Illustrator. Statistics such as Moran's I and Geary's C can be calculated to assess the level of gene expression spatial uniformity. The statistics can be compared across samples grouped by sample metadata. In spatial gene set enrichment, a p-value is calculated for each gene set to test for the existence of hotspots. The p-value is calculated using a permutation-based approach that tests against the null hypothesis of spatial uniform expression. The results can be downloaded as Excel files. Here, and across all modules, parameters of the analysis can be downloaded for future replication. The stclust and SPA-GCN methods for tissue niche detection have been implemented in SpatialGE. The two methods explicitly use the spatial coordinates of gene expression of the spots or cells to define the niches or compartments. Users can vary the weights given to the spatial information in each method. The interface allows for quick comparative analysis with the tissue images and the samples. Spatially variable genes can be detected using SpAGCN's implementation. After defining tissue niches, users can test for differentially expressed genes. Both non-spatially and spatially informed options are available. In spatial gradients, users can test whether the expression of genes is higher or lower at the interface of a tissue niche. This method does not require the definition of an interface tissue niche. Instead, it tests for significant correlations between gene expression and the spatial distance to a reference niche. The spatial gradient module can help detect interactions among regions within a tissue sample. The QR links to the publication where this method and the spatial gene set enrichment method were used to study tumor stroma interfaces in brain metastases. Two methods for cell phenotyping have been implemented into SpatialGE. For multicellular technologies such as Visium, STDconvolve can estimate contributions of different cell types to each spot. For single cell level technologies such as CosMx SMI, the in situ type method can be used. Both cases, built-in cell type reference databases are available, comprising multi-cell types from different tissue types. SpatialGE is a one-stop shop for exploratory analysis of spatial transcriptomics. It democratizes data analysis, empowering non-data scientists to explore their own data, and allows bioinformaticians to dedicate time to more advanced and customized analyses. If you are using SpatialGE and have questions or require assistance, please do not hesitate to use the contact on the left side.